Lipids show up everywhere on the MCAT, but do you actually understand what makes a fatty acid saturated? Why phospholipids form membranes? Or how structure connects to function? If these terms feel memorized instead of meaningful, that's a problem on test day. In this video, we'll break down lipids into clear, testable concepts covering fatty acid structure, key classifications, and the high-yield details the MCAT loves to ask. By the end, you'll recognize these molecules instantly and understand how to reason through any question they throw at you. Lipids are a diverse class of hydrophobic or amphipathic biomolecules that include triglycerides, phospholipids, steroids, and waxes with key structural and energy storage roles. The unifying feature is their poor solubility in water due to long hydrocarbon chains, which drives their behavior in biological systems. Triglycerides, composed of glycerol esterified to 3 fatty acids, are the body's primary energy storage form, yielding about 9 kilocalories per gram, more than twice that of carbohydrates and proteins. Phospholipids contain 2 fatty acids and a phosphate-containing head group, making them amphipathic and ideal for forming cell membranes as bilayers. Steroids, such as cholesterol, have a characteristic 4-ring structure and modulate membrane fluidity while also serving as precursors to hormones like cortisol and estrogen. Waxes are long-chain fatty acids, esterified to long-chain alcohols, and function in protection and water resistance, such as in plant cuticles. On the MCAT, you will often be asked to identify lipid classes based on structure or function, such as recognizing amphipathic molecules in membrane formation or predicting energy yield. A helpful mnemonic is TPSW: triglycerides, phospholipids, steroids, waxes, to quickly categorize lipids. This topic connects strongly to membrane structure, metabolism, and hormone signaling, so being able to distinguish these classes quickly can save time and points on test day. Fatty acids are long hydrocarbon chains with a terminal carboxylic acid group that vary in length and degree of saturation, which determines their physical and biological properties. Saturated fatty acids contain no double bonds, leading to straight chains that pack tightly and are typically solid at room temperature, like butter. Unsaturated fatty acids contain 1 or more double bonds, usually in the cis configuration, introducing kinks that prevent tight packing and make them liquid at room temperature, like olive oil. The position and number of double bonds affect melting point, membrane fluidity, and metabolic processing. A key concept for the MCAT is that cis double bonds increase fluidity, while trans fats behave more like saturated fats and are associated with health risks. Fatty acids are also named using shorthand notation, such as 18:1, indicating 18 carbons and 1 double bond, and omega numbering, which counts from the methyl end. You may be asked to predict how saturation affects membrane behavior or enzyme accessibility. Remember the mnemonic saturated equals straight equals solid to quickly recall their properties. This concept links directly to membrane fluidity, enzyme kinetics in lipid metabolism, and even cardiovascular health passages on the MCAT. Triglycerides are formed through esterification of glycerol and 3 fatty acids, and serve as the primary long-term energy storage molecules in the body. The formation involves dehydration reactions where each fatty acid forms an ester bond with one of glycerol's hydroxyl groups, releasing water. Because of their highly reduced carbon chains, triglycerides store a large amount of energy that can be released during oxidation, making them efficient for long-term storage compared to glycogen. They are stored in adipose tissue and mobilized during fasting or prolonged exercise through lipolysis, which releases free fatty acids and glycerol into circulation. On the MCAT, you might see questions asking you to identify ester bonds, predict products of hydrolysis, or compare energy yield between macromolecules. A useful way to remember structure is glycerol backbone plus 3 tails equals triglyceride, This concept also ties into beta oxidation, where fatty acids are broken down into acetyl-CoA units for entry into the citric acid cycle, linking lipids to overall cellular respiration and energy metabolism. Phospholipids are amphipathic molecules that spontaneously form bilayers in aqueous environments, creating the structural basis of cell membranes. Each phospholipid consists of a hydrophilic phosphate-containing head and two hydrophobic fatty acid tails, which drive self-assembly in water into bilayers with tails inward and heads outward. This arrangement creates a semi-permeable barrier that allows cells to maintain internal conditions distinct from the external environment. Membrane fluidity is influenced by fatty acid composition and cholesterol content, with unsaturated tails increasing fluidity and cholesterol acting as a buffer. On the MCAT, you may need to predict how temperature or lipid composition affects membrane behavior or identify molecules that can diffuse across the membrane. Small nonpolar molecules like oxygen cross easily, while charged ions require transport proteins. A helpful phrase is, "Heads love water, tails hate water," which explains bilayer formation. This topic connects closely to cell biology, including membrane proteins, signaling pathways, and transport mechanisms such as facilitated diffusion and active transport, making it a high-yield integration point. Steroids such as cholesterol have a characteristic four-ring structure and play critical roles in membrane stability and as precursors to steroid hormones. Unlike fatty acids, steroids are not composed of long chains, but instead consist of fused carbon rings that give them a rigid structure. Cholesterol is embedded within phospholipid bilayers and modulates membrane fluidity by preventing tight packing at low temperatures and restricting movement at high temperatures. It also serves as a precursor for biologically important molecules, including vitamin D, bile acids, and steroid hormones like testosterone and estrogen. On the MCAT, you may be asked to recognize the steroid structure, predict how cholesterol affects membranes, or understand hormone signaling pathways involving lipid-soluble hormones that diffuse through membranes and bind intracellular receptors. A quick recognition tip is 4 rings equals steroid. This concept ties into endocrine signaling, gene regulation, and digestion through bile salts, highlighting how one lipid class can influence multiple physiological systems tested on the exam.